Inside almost every one of your cells are tiny structures, mitochondria, that look suspiciously out of place. If you look closely, you'll see they have not one, but two membranes. The outer one matches your own cells, but the inner one is different. It's folded into complex shapes called crista and chemically resembles the membrane of an ancient bacterium. This isn't a coincidence. They even divide on their own schedule, pinching themselves in half through binary fission, just like bacteria do, completely independent of your cell's own replication cycle. And perhaps the strangest clue of all is that they contain their own separate DNA, a tiny circular chromosome completely alien to the vast linear library stored in your cell's nucleus.
This renegade genome is incredibly small, containing just 37 genes. For comparison, the nucleus holds over 20,000. 13 of these mitochondrial genes provide the blueprints for proteins essential to the final stages of cellular respiration, the very process that converts the oxygen you breathe into the energy that powers you. The other 24 genes create the machinery, the transfer RNAs and ribosomal RNAs needed to read those 13 protein recipes right there on site. But here is the puzzle. Over 1 billion years, the vast majority of the original bacterial genes have migrated to the cell's nucleus. Your nuclear DNA now holds the instructions for over 1,000 proteins that are built elsewhere and then imported back into the mitochondria. So why keep any DNA at all in these organelles? The leading hypothesis is one of control. The 13 proteins coded by mtDNA are core components of the energy-producing machinery, and producing them locally allows for rapid response to a cell's changing energy demands.
The way this separate genome is passed down is also unique. You received all of your mitochondrial DNA from one person, your mother. During fertilization, a sperm cell contributes its nucleus, but its few dozen mitochondria, located in its tail to power its journey, are left outside the egg or are actively destroyed shortly after entry. The egg cell, on the other hand, is massive and packed with hundreds of thousands of its own mitochondria. Those become the founding population for every single cell in the resulting embryo. This means you can trace an unbroken line of mitochondrial inheritance back through your mother, your grandmother, her mother, and so on for thousands of generations. This powerful genetic tool allowed researchers to trace all modern humans back to a single ancestral population in Africa, a concept popularly known as mitochondrial Eve. This isn't one specific woman, but the most recent common matrilineal ancestor from whom all living humans descend. Because it exists outside the nucleus's sophisticated DNA repair mechanisms, mitochondrial DNA mutates about 10 times faster. This high mutation rate makes it an excellent molecular clock for tracking recent evolutionary history, over thousands of years rather than millions. Every so often, a random mutation occurs, and because of maternal inheritance, it gets passed down, creating a new branch, a new haplogroup in the human family tree. Occasionally, a cell ends up with a mix of different mitochondrial DNA sequences, a state called heteroplasmy. This means within a single person, there can be a mosaic of mitochondrial genomes. Think about that. Every cell in your body is not just a part of you, it is a functioning ecosystem. It's a cooperative formed from a billion-year-old pact between two once separate life forms. This tiny circle of DNA is a living artifact, a ghost of an ancient bacterium that has become inseparable from what it means to be alive.
Monotony has grown fast. Nearly 2,700 people have watched in the last few weeks, yet less than 0.03% are subscribed. It would mean a lot if you joined by hitting subscribe. And again, thanks for watching.